Bismillahirrahmanirrahim. Assalamu alaikum. Shatin Sawati is here and welcome to Biosciences with Shatin Sawati. And today I'm going to deliver my fourth lecture from my cell biology series. And today I will discuss about the function of this role of the cell membrane and the role of different biological molecule which I discussed in my previous lecture like cholesterol and phospholipids and proteins and many more. And next I will discuss in this lecture the most important biological molecule which are conjugated molecule glycolipids and glycoproteins. So now you can see here in the figure all the biological molecule and like phospholipid bilayer membrane and many peripheral and integral proteins, cholesterol and many more. So now I'm going to discuss about the role of the biological molecule. All biological molecule like phospholipids, proteins, carbohydrate, cholesterol, they play a very important role in the cell membrane function. All the functions of the cell membrane actually carried out by all these biological molecules. So must watch my first lecture about the biochemistry of the cell membrane, so it will be easy for you people to understand this lecture. So now I'm going to start phospholipids. First of all, phospholipids. Phospholipids, as I have discussed, present in the form of the lipid bilayer, are critical component of the cell membrane. The lipid bilayer act as a barrier to the passage of the molecules and ions into and out of the cell. However, an important function of the cell membrane is to allow selective passage of certain substances into and out of the cell. So, the phospholipids actually, there are four types of phospholipids I have discussed. So, must watch my first lecture about the biochemistry of the cell membrane. So, this will be clear upon you people. So, I'm now here discuss again all these things. So, phospholipids not only provide the fluidity to the cell membrane but play a very important role and act as a selective barrier and allow selective molecule to pass through the cell membrane. This is accompanied by now the second one thing. First one is the phospholipid. Second one is the protein. Protein molecules present in different form. I will show you conjugated molecules are also present here in the cell membrane. So the proteins which are in peripheral proteins and integral proteins play a very important role for the movement of the certain specific ions and molecule across the cell membrane. I will show you through diagram. Now the third one is the carbohydrates. Many membrane proteins also contain and also attached with the carbohydrates. On the outside are the outer leaflet of the cell membrane and as well as on the inner leaflet of the cell membrane, because cell membrane consists of two layers, lipid bilayer. Allow it to form the hydrogen bonds with the water. The most important function of carbohydrate is to form the hydrogen bond with the water. So these molecules, phospholipids, proteins, carbohydrates performing different function in the lipid bilayer. Okay, now you can see here all the molecules which are, this is the lipid bilayer and you can see here the sea of lipid in which the proteins float, which is the fluid mosaic model. You can see the peripheral proteins and the oligosaccharide means carbohydrate chains, and you can see here the channel proteins which help in the transport of different ions, because cell membrane which is selective permeable membrane and the lipid bilayer membrane. This lipid bilayer membrane allow selective movement of the substances through it. So this is the lipid and lipid bilayer, and lipids are non-polar. As I have discussed early, that the lipids are the non-polar, and this non-polar membrane allow the movement of the non-polar substances easily. Small size and non-polar substances, and those substances which dissolve in the lipids, can pass through the cell membrane easily. Is it clear? The first one thing, that the small size, and the second one thing, the molecules which dissolve in the lipids, phospholipids, can easily transport through the cell membrane. Other ions, large molecules pass through the channels, protein pumps used for that purpose. Is it clear? Now, the role of cholesterol. You can see here the small cholesterol molecules are also present. This is the phospholipid bilayer. These are the carbohydrates chains, and these are the integral proteins. So this is the peripheral protein. You can see each and everything clearly. So cholesterol has a role in membrane fluidity. Actually, cholesterol provide the stability to the cell membrane, fluidity as well as stability. But its most important function is in reducing the permeability of the cell membrane. Cholesterol help to restrict the passage of the molecules by what? By the increasing the packing of the phospholipids. So it provides the stability to the cell membrane by packing of what? Phospholipids. It also help in the fluidity. It also help in the permeability. Is it clear? Means the reducing the permeability. Is it clear? Now, glycolipids and glycoproteins, what are they? These are actually the conjugated molecules. What are conjugated molecules? Now students confused about that. Glycolipids and glycoproteins. Glycoprotein means they are made up of the carbohydrate, glucose, or other oligosaccharides. So glyco is the word from glucose, from carbohydrate, and protein. So two different molecules combine and form the one molecule, which is the conjugated molecule. So glycoprotein, which is the combination of two different biological molecule, one is protein and other is the carbohydrate. Similarly, the glycolipids, carbohydrate plus lipid, two different categories of molecules combine and produces the conjugated molecule. So glycoproteins and glycolipids concept is clear, and you can see here in the figure the clear image of that, glycolipids and glycoproteins. Now here you can see the chemistry of the glycolipids, and this is the sugar chain which combine with the lipid phospholipids and produces the glycolipid. And this is the chemistry of that glycolipid. Is it clear? Now I'm moving towards the glycoproteins. Glycoproteins, you can see here, this is the protein, integral protein of the cell membrane, and to which the glucose chain or carbohydrate chain is attached. So the molecule is the glycoprotein. Now glycoprotein exists in two forms, O-linked and N-linked. O-linked and N-linked, as you can see here, depends upon the bond. You can see here the molecule glyco, this is the glucose or carbohydrate molecule, and this is the protein residue. So the proteins attach or bond with the carbohydrate at O, And here you can see the clear difference. This is the amino group of that protein, N, NH amino group. So the bond depending upon the bond, there are two types of the glycolipid, O-linked and N-linked. The concept clear now? Now I'm moving towards the role of glycolipid. Very important. Like you can see the role of the phospholipids, you can see the roles of the proteins and cholesterol. Now you are here learning about the role of the glycolipids and glycoproteins. They form hydrogen bonds with the water molecule, like the carbohydrates form the hydrogen bond with the water molecule surrounding. You know that the cell inner leaflet and outer leaflet, cell membrane inner and outer leaflet is surrounding by the aqueous compartment, and thus help to stabilize membrane structure. Like the cholesterol, like phospholipid, glycoproteins and glycolipids help in the stability of the cell membrane. They are used as a receptor molecule. This is very important. These glycolipids and glycoprotein act as a receptor for hormones, for neurotransmitter, and many more things, to trigger a series of chemical reaction within the cell itself. They act as a receptor. They receive substance because cell membrane is selectively permeable membrane. As you know that the cell membrane is lipid bilayer, and this lipid bilayer is selectively permeable membrane. Allow the very small molecule or those molecules easily which dissolve in the lipid bilayer, lipids. So the larger molecules, larger proteins and other large molecule can pass through the integral proteins or many other types of the proteins which are present in the cell membrane. So glycoprotein in the cell membrane are very important for cell to cell recognition and immunity. Now they help in the recognizing of the cell and also provide the immunity. Very important role play in immunity. Now glycoproteins help in the cross-linking cells, help in the linking of the cells and proteins to add strength and stability to a tissue. There are almost very important 4 functions. Number 1, the stability of the cell membrane. Number 2, as a receptor. Number 3, play a very important role. Cell to cell recognition, mean recognizing of the cells, and immunity. And then about the strength and stability of the tissues because of the cross-linking. They join the cell, they link the neighboring cells with each other, and then cell, you know that the cells group together form the tissues. So the tissue stability is because of the glycoproteins and glycolipids, and then you know that the tissues group together form the organs. So the all cells connected with each other, linked with each other because of these glycolipids and glycoproteins. They play a very important role. So must keep it in your mind. All the biochemistry of the cell membrane. Actually, this biochemistry or biological molecule help you to understand actual function of the cell membrane, and you can answer each and every question related to the cell membrane if you have a concept of the biochemistry of the cell membrane. Is it clear now? This is very important and very interesting too. I hope so you are enjoying. Now, the transport across the cell membrane. Okay, ji. Now, you can see here, this is the cell membrane, and you can see here different types of the movement across the cell membrane. Across the cell membrane, you can see the passive transport as well as the active transport. Now, what is the difference between the passive transport and active transport? Passive transport is that transport which easily allow the movement of the molecule across the membrane without the expenditure of the or using any energy in the form of the ATP. Simple movement like diffusion, facilitated diffusion, etc. So the second one is the active transport. Many large ion which across the cell membrane, like sodium, hydrogen, potassium, many more, when they move against their concentration gradient, means from lower to higher concentration. Against concentration gradient means the movement of the molecule from the lower concentration towards the higher concentration. If the just suppose I'm giving you an example of the sodium ion or potassium ions or hydrogen ions. If these ions are greater in concentration outside of the cell and less in concentration, this is the outer side of the cell, this is the inner side. Just suppose if you say that the concentration of the ion is greater outside of the cell, or you can say that if the inside of the cell, if this concentration is greater outside of the cell and the sodium ion concentration lower inside of the cell, so if the movement of the ion from this low concentration to the higher concentration of sodium ion require energy, and that energy is provided by the breakdown of the ATP. ATP breakdown into ADP and phosphate, and energy is released, and that energy is used to push the sodium ion or potassium ion or hydrogen ion from lower concentration to higher concentration. But in the simple passive diffusion, what will happen? Passive transport, simple diffusion, and passive transport always occur from higher concentration to lower concentration, but the active transport occur against the concentration gradient, means from lower concentration to higher concentration. Now you can see the clear difference between the passive transport along the cell membrane and active transport along the cell membrane. There is another, like this is a simple diffusion of the oxygen molecule. Now you can see here the facilitated diffusion. Now what is facilitated diffusion? Facilitated diffusion is that type. Now you can see here the sodium ion concentration move from higher concentration towards the lower concentration. This is also the simple diffusion process. But this is the what? This is the facilitated diffusion, in which the like glucose molecule, large molecule, other molecules which are larger in size, they cannot pass through the plasma membrane, because plasma membrane is selective permeable membrane. And you also familiar that this is the lipid bilayer. So small size and those molecules which dissolve in the lipids can pass through it. So the glucose molecule, which is an organic molecule large in size, this can pass through the proteins, means help with these proteins help in the movement of the glucose molecule or other large molecule. So this sort of diffusion is known as facilitated diffusion. That facilitated diffusion help are facilitated with the proteins. Proteins carry the large molecule across the cell membrane. Is it clear? So the last one, active transport, which is against the concentration from lower to higher concentration and need energy. The concept is clear now? Okay. Now you can see here there are different mechanisms like endocytosis, exocytosis. I'm giving you just a concept because students confused about the endocytosis, exocytosis, and then pinocytosis and phagocytosis. I'm here giving you the concept. Now the concept clear about the passive transport and active transport, why the large molecules cannot pass through the cell membrane, why do the small molecules and non-polar substances can easily pass through the cell membrane. The reason is that the lipid bilayer, lipids, only those substances which are small in size and dissolve in the lipids can pass through the cell membrane easily. So here this is the endocytosis mechanism. In this process you can see the endo means in, cytosis means cell, the movement of the cytosis, movement of the substances, any food particle or any other thing from the outer environment, means the extracellular environment, to the inner environment of the cell inside the cytoplasm. So this is the endocytosis. You can see here the cell membrane form the pouch-like structure. Then this pouch later on separate from the cell membrane and exist in the form of the vesicle. containing the food material or any other proteins or maybe any other receptor, maybe any the viruses, bacteria. All these things move by the mechanism of endocytosis. Okay, now exocytosis. This is that mechanism in which the cell excrete or secrete both. You can see here the substances which are present inside the cell, now they have to move outside of the cell, means the extracellular environment. Now exo means out, so the movement of the substances, you can see here, this is the vesicle which carry the substance which is now move outside. This vesicle come and attach with the cell membrane, and the enzymes help in the dissolve the cell membrane, dissolve here, and a pore is created here, and the vesicle then all the material which is present in that vesicle, then after fusion what will happen, fuse with the cell membrane, and all the material come outside of the cell, maybe protein, maybe any other thing. So what is the mechanism of endocytosis and exocytosis? This is clear now, how the vesicle produce in case of the endocytosis, and how the vesicle is produced, and the substances move outside of the cell by movement and attachment with the cell membrane, and the enzyme then dissolve that cell membrane, and the vesicles the substance present in the vesicles come outside, and the vesicle become empty. Now this is the clear most concept about the pinocytosis, phagocytosis, and receptor mediated endocytosis. Now what is the phagocytosis? Now I'm giving you the concept. Phago means the eating process of the cell, in which the cell eat large particles. Is it clear? Solid particles, large particles eaten by the cell, and the mechanism is same, endocytosis. This is endocytosis. Phagocytosis, pinocytosis, these are all endocytosis, in which in phagocytosis what will happen? Large molecules are taken up by the cell, inside of the cell. This is the what? Phagocytosis. Pinocytosis. Simple, you can say liquid molecule. Liquid intake of the cell is the pinocytosis. This is the type of endocytosis. The third one is the receptor mediated endocytosis. What is receptor mediated? Means you can see here, these are the glycoproteins which act as a receptor. These receptors help in the selection of the, like, hormones come, like neurotransmitter come, like other microorganisms maybe come. So what will happen? These receptors recognize and take it inside of the cell. So such sort of the movement is known as receptor mediated endocytosis. Is it clear? The concept of this is endocytosis, types of endocytosis, in which phagocytosis, which is the eating of large particles or solid particles. Pinocytosis, drinking process of the cell, in which the cell intake the liquid material, and receptor mediated, in which the receptors, which are glycolipids and glycoproteins, which are present on the surface of the cell membrane, and these help in the recognition and then movement of these substances into the cell. So such sort of movement is known as receptor mediated endocytosis. And you can see here the coated vesicles. Why? Now simple vesicles, these are the coated vesicles. The reason is that these vesicles are coated with different proteins. Like you can say that the virus enter into the cell, so the receptors first receive and then enter, and the vesicle is produced by the infolding of the cell membrane, and then small proteins attached to that vesicle. So this is what, and in these coated vesicles actually help in the movement of the substances like proteins or many other things from one part of the cell to the other. Means the movement of the between the distinct part of the cell occur through the coated vesicles. and these proteins also help in the recognition of that site. These proteins help to recognize where that particle, where that protein uh, have to reach. Is it clear now? I hope so. The concept is now clear. Now, this is the whole process which I have explained one by one: phagocytosis, pinocytosis, and endocytosis, exocyte, each and everything. Endocytosis, which may be pinocytosis, which may be phagocytosis, which may be receptor mediated endocytosis. So all these you can see here. Now the concept is clear upon you people about the mechanism of the transport, and as well as the all the biological molecule role in the cell membrane. And you can see here, I will explain it in my next lecture, that how the eating of the, or you can say the autophagy occur. You can see here the mitochondria, which is now ruptured or destroyed, is taken up by the lysosomes. I will discuss it later on, but here I am just giving you the concept of cell membrane function. So don't be confused about this mechanism. I will explain it in my next lecture, inshallah. So keep watching my lectures, like my videos, share my video. Thank you very much. I will be back tomorrow. Allah Hafiz.